Hello guys, welcome to 13 Universal Learning Beyond Barriers. So we are going to also continue on A-level Caie biology, and this is schedule. So we're going to cell membrane and transport series 2. If you have not listened, watch series 1, go watch it. Okay, this is where we solve questions to give you tips on how to crack this concept, the questions using the concept, how to apply the learned concept. Okay, so come with me, let's dive into the questions. Number one, okay, it says single-celled animals. Okay, single-celled, that is unicellular animals that live in fresh water. Fresh waters, they have less ions compared to the sea water, have a vacuole that contracts regularly, so they have that contract regularly to remove excess water. Single-celled plants that live in fresh water do not have a similar vacuole. Which statement explains why these animals need this vacuole but plants do not? Okay, why is the plants do not need vacuole? Animals need vacuole, plants do not. Okay, let's look at it. Pause this video, check whether I can solve it. Okay, let's dive in. Is A the correct answer? Let's see. Plant cell cytoplasm and animal cell cytoplasm both have a lower water potential than fresh water. Okay, yes, that is why water moves into them. But the question is, why is it that animals need vacuole but plants do not? It's not why is it that water moves into them. Okay, A is not correct. B says plant cell sap has the same water potential as freshwater. Animal cytoplasm has a lower water potential than fresh water. It doesn't really mean that. It doesn't explain this. Okay, it doesn't explain why this needs vacuole and plants do not. Okay, all right, because if plant cells have higher water potential, is that okay? Because if it has the same water potential, there's no way the net movement of water will be inside plant cell. So that means that plant cell of course has lower water potential because it's inside fresh water. Okay, so B is cancelled, eliminated. C says plant cell walls are impermeable to water. Animal cell surface membranes are permeable to water. Plant cell walls are not impermeable. They are not impermeable because cell walls are made of cellulose. Cellulose are polar, and the microfibrils of cellulose have pores that allow water movement. Water moves through the cell wall by mass flow, okay, as a body of liquid, so it's not impermeable. Okay, sorry, it's not impermeable. Yes, so that leaves us with D. Plant cell walls restrict entry of water. Animal cell walls allow, animal cell membranes allow the entry of water. So plant cell wall restrict. How does it restrict, guys? Tell me, how does it restrict? Remember, guys, remember, just want to draw this. Okay, if this is to be a plant cell, all right, follow me. Now the plant cell has cell wall. Don't forget. All right, this is cell wall made of cellulose. Okay, it also has large vacuole. Just don't talk about that. Okay, when water enter, water exert pressure. Look at this. This is pressure against the cell wall. Okay, that is cortical pressure. But cell wall also, of course, it says so. In a way, cell wall is also exerting pressure against water molecules. Do you get? Yeah, that is called osmotic pressure. All right, so because of that, all right, because of osmotic pressure, the plant cell will restrict the entry of water when it gets to a point where the amount of osmotic pressure is equal to the total pressure. Then that reduces inflow of water. The pressure prevents the movement, is that okay, of water. Okay, I think that is the answer. That is D. Okay, you can ask us question. That is D. Let's go to the next one. Say increasing which type of bond helps to increase the fluidity of the cell surface membrane. Okay, not that bond, that, okay, this, okay, the bond, because when we talk about the fluidity of cell surface membrane, we're talking about hydrocarbon chain, okay, and in the tail of phospholipid molecule. Do you get? And then we have carbon to carbon double bond. Okay, this is unsaturated. So the more unsaturated, the more fluid. Okay, carbon to carbon. This is double bond, unsaturated. Unsaturated in the sense that we have only two hydrogen that can attach this carbon atoms here. Okay, so less hydrogen atoms, double bond means unsaturated, and then that increases the fluidity. All right, as fluidity increases, as level of unsaturation increases, fluidity increase. Also, as the length of the tail of phospholipid shortens, fluidity also increases. Look at this question. The diagram shows three routes, X, Y, and Z, through which substances can pass across a cell surface membrane. Three routes. So look at that. This is a three route. One is through the phospholipid bilayer. This molecule X must be lipid soluble for it to be able to pass through. It must be small and lipid soluble. If it's not lipid soluble, the tails will repel it, because these are the tails they call hydrophobic core. They repel polar molecules. Now Y may also be lipid soluble, but CZ, okay, Z is also lipid soluble. No, Y is not lipid soluble, okay, because passing through protein. Z also is passing through protein, so Z and Y, what more likely water soluble polar. Is that okay? So they are passing. This one is passing through the hydrophilic channel of the hydrophilic passageway of channel protein. All right, look at this. They say which correctly shows the route for the vitamin D. Sorry for the background noise. I'm in a school environment. Okay. Which correctly shows the routes for vitamin D, which is fat soluble, and vitamin C, which is water soluble. So vitamin D is fat soluble. It's going to pass through X. So C, which is water soluble, is it going to be Y or Z? That's the question. Is it going to be Y or Z? All right, and we have, so you know that D must be X. Okay, so we have BC. So which one is the correct answer? Now, if you look, you see that this molecule is actually moving down concentration gradient Z, and it's passing through this channel. This is channel protein. Okay, this is a transport protein. Okay, this doesn't look like it doesn't look like a carrier protein. There's no binding site shown. Okay, even though it's as the molecule is passing through, but it doesn't show binding site. Okay, but this is a channel protein. It has hydrophilic passageway through which molecule passes through, and this molecule is moving from outside to inside. Is possibly moving down concentration gradient. Okay, it's moving vitamin C, because the cell does not make vitamin C, the cell uses it. All right, so the answer is X and Z. That's the most suitable answer to this. Okay, let's go to the next question. All right, look at this question. What is the width of the cell surface membrane? Now, this is a known fact. Please don't forget it. The width is usually between 5 to 10 nanometers. It's a fact. All right, 5 to 10 nanometers. 5 to 10 nanometers. All right, so just note that. Number 5, some functions of molecules found in cell surface membranes are listed. We have cell recognition, ion transport, maintaining fluidity, mechanical stability. What are functions of cholesterol? Okay, what are functions of cholesterol? Okay, let's walk through it. Cell recognition is done by glycolipid. Don't forget it. Glycolipid, glycoprotein, and protein. Okay. Let me just show you something. I don't know what you should have seen it before. Okay, glycolipid is just phospholipid molecule. See, phospholipid molecule. This is phospholipid. The tail, the head, and the tail. Now, when it's attached to carbohydrate chain, it becomes glycolipid. Okay, that's for recognition. Now, glycoprotein. This is a protein, right? This molecule. Now, plus glucose chain makes a glycoprotein. All right, this is just protein. Okay, 3 of them, glycolipid. This is, all right, glycolipid. glycoprotein, protein, they both are involved in cell recognition. They act as antigen. Ion transport is for protein alone. Okay, channel protein and carrier protein are involved in ion transport. Maintaining fluidity. Okay, yes, that's where cholesterol comes. Cholesterol helps to maintain fluidity of the membrane. So at high temperature, cholesterol will form bonds with phospholipid molecules so that they can become less fluid. Okay, at low temperature, it will create kinks or gaps between them so that they are not frozen. Okay, and also is involved in mechanical stability. So cholesterol is involved in maintaining fluidity and mechanical stability. Don't forget it. Okay, so the answer to this question, of course, is delta. Now they say the enzyme lactase is found in the membrane of epithelial cells lining the small intestine. The enzyme is formed by a single polypeptide that folds to give three regions. Okay, all right, so let's see. An active site with free amino acid group outside the cell, a short section inside the membrane, a short section inside the cell. What type of amino acid will be found in each of the three regions? So look at it. Look at pause this video. I want you to tell me the answer to this question. Tell me the answer to this question. So if I draw something like, watch this, guys. If this is a cell, okay, this part is outside. Outside there is water there. This is inside. There is water there. All right, inside and outside. Outside. So any amino acid that's facing outside must have water. It must be hydrophilic. Sorry, it must be hydrophilic. The one inside must be hydrophilic, but the ones inside the membrane must be hydrophobic because of hydrophobic core. Remember hydrophobic core of this phospholipid bilayer. Okay, the hydrophobic core repels polar molecules. So any molecule that be inside there must be hydrophobic. The amino acids that will be there must be hydrophobic because that core repels them. It repels polar molecules. So any amino acid that will be found around here, this is inside the membrane, must be hydrophobic. So outside is hydrophilic. Which option? A and B. So C and D eliminated. Inside must be hydrophobic. Okay, two of them still goes. Now that's inside the membrane. Sorry, inside the membrane, inside this membrane must be hydrophobic. All right, because of the phospholipid bilayer, and the inside that is here is also hydrophilic because it contains water. Okay, so that happens to be option A. Is that okay? If you have a question, you can let us know. Number 7. Which two substances maintain the fluidity of the cell membrane? Of course, you should have known that now. Tell me the answer. Cholesterol and phospholipid bilayer. Okay, glycolipid, antigen, cell adhesion, the cell recognition, cell adhesion, glycolipid, glycoprotein, cell adhesion, cell recognition, cell signaling. All right. Okay, phospholipid and cholesterol, they help to maintain fluidity and they prevent the passage of polar molecules. Is that okay? Let's go to, so that's D. Let's go to the next question. It says the membrane proteins are called peripheral. See, if they are temporarily attached to the membrane, or integral if they are permanently attached to the membrane. Integral proteins are described as intrinsic, that's embedded, if they extend across the whole bilayer, and extrinsic if they are found only in one side of the bilayer. How would you describe a channel protein? How would you describe a channel protein? Read this question and attempt it. How would you describe a channel protein? Let me paint this picture for you guys. Look at what I'm trying to say. Come with me. Let's reason. All right, this question out. Okay, this is, of course, this is the monolayer of the phospholipid bilayer. You can see that seed phospholipid with hydrophilic head and hydrophobic tail. Okay, so we have something like this. This is the tail. That's the tail. Okay, that's the tail. All right, now this is it. Okay, without wasting much time, Okay, some proteins can be found here, right? They don't span across, right? So, but you see, we're interested in the one that spans across. All right, so look at this. Yeah, sorry, I'm trying to draw this, but look at this. This is channel protein like this. Okay, that's it. All right, channel protein. Okay, so you can see spans across bilayer, right? Spans across bilayer. So the question is, this integral protein are described as intrinsic if this is extended across the whole bilayer. So this is channel protein is extended, so it is intrinsic. Okay, all right. And they are permanently attached, so they're both integral and intrinsic. They are permanently attached because they are embedded in the membrane. This is channel protein. Embedded is embedded in the membrane, but they also span across from one bilayer to another. Can you see? This is intrinsic, it is integral, but extrinsic. It doesn't span across. Okay, but cell membrane, channel protein is both integral and intrinsic. It spans across the bilayer. So it is both integral and what? Intrinsic. Amazing. The diagram shows apparatus set up to investigate the effect of changing the concentration of glucose in the surrounding solutions on the movement of molecules through a selectively permeable membrane. Okay, in 15 minutes. That's Vatican tubing. The net diffusion of water. Okay, these are the things they were saying, and they're not asking you what's the option. Pause the video and try it. Okay, pause it. The diagram, this diagram, shows apparatus set up to investigate the effect of changing the concentration of glucose in the surrounding solutions on the movement of molecules through a selectively permeable membrane. So they are changing the concentration of glucose in this surrounding solution, this surrounding solution, okay? And that they are actually checking the effect. So they're increasing the concentration from 1% to 10%. Remember, the question says, as the concentration of glucose solution in the surrounding increases, which statements are correct? Okay, which statements are correct? Now, number one says net diffusion of water increases. No, see, if you start increasing the concentration of glucose here, the water potential, because we are adding glucose from 1%, we add 2%, 3%, 4%. So you see, as the solute concentration is increasing, water potential will decrease. The water potential between here and there will decrease. So so net diffusion of water will not increase, it will decrease, because we are making the water potential gradient less by increasing solute concentration here. All right, we are decreasing the amount of water available to move inside. Do you get? 2 says glucose molecule reach an equilibrium quicker. Yes, as you keep adding 1%, 2%, 3%, before you know it, once you get to 10%, all right, here is 10% and there is 10%. That creates an equilibrium. By increasing it, we're going to reach equilibrium quicker. Instead of allowing glucose to diffuse out from here to here, we just create equilibrium by adding glucose inside here. All right, so this is correct. That is wrong. 3 says there is less change in the volume of surrounding solutions. Yes, there's less change. Okay, why? Because there is less movement of water. Water supposedly should move from the surrounding solution into the Vatican tubing. All right, at low concentration of glucose outside, at low concentration, they're going to be high water potential than this. For example, if I have 1% here and this 10, this has high water potential, water is supposed to move in. Okay, but as I continue to increase it, the amount of water moving will be less, and therefore the amount of the volume of this outside solution that decreases will also be less. All right, it will be less because less amount of water is increasing, so it's moving inside. Why? Because I'm increasing the concentration, all right, from 0 to 10. All right, look at this. Net diffusion of glucose increases, all right, it will not increase. all right, because I am making the concentration gradient for glucose less steep. I am increasing here with concentrate with glucose, all right. So the amount of glucose moving from here to here will decrease because I'm already increasing here with glucose concentration. Okay, so 2 and 3 seem to be correct. What do you think? What do you think? Give me what you think. All right, so 2 and 3. All right, which type of cell has a large number of glycoproteins on the cell surface membrane? On the cell surface membrane, large number of glycoproteins. So which one is involved with cell recognition or cell signaling? Which of them? All right, ciliated cell is involved in movement, so it should have more mitochondria. All right, it is not involved with of course much with cell signaling or cell recognition. Goblet cells secrete mucus. All right, and lymphocytes are white blood cells, so they are involved in immune response. There's an immune response called signaling, of course, clonal selection. So lymphocytes have receptors that allow them to bind to the antigens of pathogens. So this should have glycoproteins. Red blood cells just transport oxygen. All right. So among 4 of them, lymphocyte should involve more in have more glycoproteins, which are the receptors on them with which they interact or bind to the antigen on pathogens. What do you think is the answer, guys? Lymphocyte, of course. Thank you, guys. Please kindly leave your questions. Of course, if you have questions, comment, subscribe. Make sure you subscribe to our videos and make sure you share, like, and ask a question. I want to especially thank you for going this far with me. You know, just tell us what you want. We're there for you. Other subjects are coming. Just want you to know that other subjects are coming. Science, art, they are coming. All right. Thank you. See you next class. Remember, this is schedule. Bye.